It's not good to me. It's not good to me.
I think that you only work I don't know. Uber works at 4 a.m. but they'll charge you it.
She's kind of petty about it.
I guess that.
I'll keep bringing that up.
Pardon me? Well, I mean, when you, I'll tell you. Okay. Anybody have any questions? See, we're getting a little movement around here. You're throwing me in a little loop here. Natalie was able to post the first recording lecture on Moodle. There's just one of the pages that says lecture recordings. I forgot to record the lecture the other day, so I'm going to ask Sarah and some other people maybe sitting around here. If you don't see me turn on the recorder, give me a quick reminder. But I'll try to do that routinely. So we'll go for regular period about 10 till, take a 10-minute break, and then we'll do a little bit of lab orientation. And we should finish well before the 10 to 11 time when lab ends. Okay, any questions? Okay, so what we've accomplished so far, hopefully, is given you some kind of idea of the scope of relevance that microorganisms have. And I hope I broke through a little bit of the barrier of this common misconception that microorganisms are all bad germs. That is an important part of what they do, and one of the things we're going to be studying is pathogenic microorganisms, and particularly the body's response to those when we talk about immunology. But by far, the beneficial things and the essential things that microorganisms do outweigh that particular negative aspect. So that sets up a little bit of kind of the context of why we bother spending a semester digging into these little single-cell prokaryotic organisms. What I want to do now is talk a little bit about history of microbiology, and particularly the evolution of the science of microbiology. And as we talk about that, we're going to fold in a couple of important themes. Those two themes we're going to talk about is spontaneous generation, does it occur or not occur? And two, what causes contagious disease? Those were two questions that were at the center of the evolution of the science of microbiology. And as those questions were investigated, microbiology developed from a curiosity to a really legitimate science. And one of the things I hope you will understand is how the investigation of those two ideas, spontaneous generation and contagious disease, led to the establishment and the legitimacy of microbiology as a science. So at the beginning of the week, we talked about the fact that one of the things that characterizes microorganisms is that they're small. Okay? And if we start looking at a diagram right here of various types of living things, and I've got this expanded just a little bit, it's hard to see the top right there. So we look at things that can be seen with the naked eye, things that can be seen only with the use of a standard light microscope, and then things that are submicroscopic that require advanced technologies like electron microscopes, both scanning and transmission electron microscopes, and even some new microscopes, things like the scanning tunneling electron microscope, which can really get us down into the molecular and even close to the atomic level. So we've got this kind of whole range of sizes of organisms, and one of the things I want you to understand is that we're down here in this area where to be able to visualize the organisms that we're studying at the very least we need a good light microscope. And in some cases, for some of the things we're going to study, you really almost need an electron microscope to be able to visualize these things. Okay? So we're in a different realm of measurement when we start talking about these microscopic and submicroscopic entities. For example, the range of the light microscope typically is used to look at things that are somewhere in the vicinity of 1 micrometer in size up to maybe as large as 100 micrometers in size. Do you all know what a micrometer is? Or a micron? What is it in relationship to a meter?
10 to the negative 6.
10 to the minus 6 meters. Okay? Small. Okay? One of the things we're going to be doing in lab next week is making sure that you are comfortable with the use of our compound light microscopes, the one sitting over in the cabinet over here, and particularly with the oil immersion lens. That is our highest magnification lens. That will give us the capability to get 1,000 total magnification. A 10x ocular and a 100x oil immersion lens gives us a total magnification of 1,000x. But one of the things we're also going to talk about is that when we deal with microscopy, magnification is only part of the game. So we're also going to talk about the phenomena of resolution and resolving power and make sure you understand what that is and why putting oil on that oil immersion lens is able to increase the resolving power of that particular objective. We're going to look at some representative types of organisms, so you get used to kind of looking at what the size is of these organisms. And we're going to do some measurements. And the measurements will involve standardizing a little device that's in the eyepiece of our microscope called the ocular micrometer, so we can be able to accurately measure microorganisms. That's going to be important when you deal with your unknown. Particularly certain types of unknowns, size measurements will really be important. And for some bacillus species, the size of their endospores can be very important. So we really have to be able to be pretty accurate in measuring microorganisms. So that's something that we're going to be doing in lab next week. So you really should get kind of comfortable with this idea of we're working down here in this micron range, this micrometer range. We're typically not talking about things that are millimeters or meters in size. Well, where does the history of microbiology trace back to? Well, it's really hard to study things if you don't know they exist. Okay? And prior to the development of the microscope, nobody really knew about all these single-celled critters that were found in the air, in the water, in food, etc. So really, much of the history of the development of microbiology as a science is paralleled with the history of the development of the microscope. Now, it's really hard to absolutely say this is the person that developed the microscope. And there is a lot of actually disagreement if you talk to science historians about who should really be given credit for that particular accomplishment. Because it was huge. It opened up this whole new microscopic world, and people started to understand that there were all these different organisms out there that they didn't have any idea that existed prior to that. Okay. Laptop. Give me a second.
I was so upset when I was
That's okay. I'm not getting this way. I'm not getting this.
You like that?
What?
You watch People Riders?
Yes,
I love it.
It's so good.
Yeah, they're shooting season 4.
Yeah. And then the seasons are only 6 episodes, so it's super easy to watch. So good. Who do you like the most?
Who do I like the most?
I like Tom Shelby.
So, I like John.
Yeah, I do too.
Savage.
I hate Arthur.
He's a psychopath.
Yeah, not you. Yeah, he's crazy.
And now he's like, well, at the end he's like super like Christian and stuff.
Yeah, why?
I hated the cop.
Yes,
It was
he's
so
god
annoying. It
awful.
was so annoying.
Yeah, he just like needed to die.
But his accent was so stupid.
Yeah.
Right,
There's
thanks for asking about it.
There's another cop.
I think
I
we
love
liked
not
it.
watching it.
I think we loved it.
I'm so sorry guys.
It was.
Maybe I need somebody.
You see the cop
I'll
next.
look at my.
I sent him this email, and he CC'd on, when you email the accent, and said like, we didn't have the note.
He gets shot. Or I don't want to know. I think he dies, but he comes back. It was a little bit.
Yeah.
But he gets shot again.
Yeah. Probably like more in like why were you doing it? Why do you expect to get these sort of results? Probably asking questions like that, not just that. He's like most undergraduate programs like that. Like, here, you just want to know if you understand it. Yeah, that's what they did to me too.
Do you have a license?
Because
I'm calling the TRs.
Then he went up to me like telling articles. He's like, I expect you to have read the next 2 weeks. I was like,
That's right. It's up to their interpretation.
I read like
They don't
it
call
took
it
me longer than 2 weeks, but I read them.
I don't even have a pull down here.
Citizen arrest.
Well.
Honestly, you will only be fine because the chance of getting to find a slave.
Back to that.
Dr. Rowe.
It does, so. That's easy that.
Okay, so most people that deal with history of science give the credit for kind of the development of the microscope to Anton von Leeuwenhoek. And look at the time frame, 1632 to 1723. So until this period of time when microscopes first started being used, nobody knew that microorganisms exist, and so obviously microbiology as a science couldn't have predated this particular discovery. Von Leeuwenhoek was a Dutch linen merchant. He wasn't a formally trained scientist. And one of the things that he did was he developed little handheld optical devices. And the reason that he did that was that he was looking for flaws in fine linens. And so he needed some kind of magnifying thing to see little flaws that might occur in the linens that he bought and sold. But he was also a pretty curious guy. And he did a lot of interesting experiments with his little handheld microscopes. And one of the things that he was interested in and investigated was he liked to cook with pepper. And one of the things that he was interested in is what makes pepper hot or spicy. And he was hoping that if he could look at pepper in greater detail with greater magnification, he could maybe see something about the peppercorns, for example, that would suggest why they had that particular property. Sound familiar? Structure determines function. Okay. So he ground up some peppercorns and he emulsified them in water and observed them under these little handheld microscopes that he developed. Amazingly, he was able to get a magnification with these microscopes of as much as 300-fold. And here's an example of the von Leeuwenhoek microscope. And it's a pretty primitive device if you look at it. And obviously it just has a little kind of stage, a single lens. It's not a compound microscope. And a little focus screw to kind of move the little metallic stage up and down. And he just held it up to his eye and put samples on the little sample holder and was able to then look at things in greater detail than anyone had ever seen before. Well, when he took some of his peppercorn preparations that had been in water for a while, that had been sitting around for a while, and looked at them under the microscope, he saw something really interesting. And that is, he saw all kinds of little microscopic organisms, and these are some of his drawings. And this just blew him away, because these are things no one had ever seen before. If you looked at the water looked particularly clear. It didn't look like there was anything in it. But submicroscopic examination indicated all these little tiny organisms, some of which were bacteria, and he was the first person that really documented the existence of these particular organisms. He referred to them as small animals or animalcules. He presented his findings to the Royal Society of London, which was the preeminent scientific organization of the time. Isaac Newton, all kinds of famous scientists were part of the Royal Society. And they were just shocked. And once people got wind of this particular idea that there were all these little microscopic critters living in water and later found in air and in food and things, it just opened up this whole new world of discovery. But at the time, it was still really just kind of a curiosity. Okay, there are these little things there, but what are they doing? Do they have any real significance? They were so tiny, I think most people believe that really they didn't have much significance at all. Well, as the microscope improved, we saw more and more detail about microscopic organisms, particularly in the 1950s when the electron microscope was developed, and that opened up the whole world of understanding that there were two fundamental types of cells, prokaryotic cells and eukaryotic cells. But over that period of, almost 300 years, the microscope made incremental improvements, and with those improvements we learned a little bit more about the nature of microorganisms. But again, the microorganisms were just kind of a curiosity. What was their significance? What were they doing? We don't really consider microorganisms, or microbiology, as becoming a legitimate science until about the mid-1800s. The mid-1800s is sometimes referred to as the golden age of microbiology. And that's when it really first started to take off, and people really first started to appreciate the fact that man, this was something significant. This science really was important, and these little tiny organisms were very important. So we've known about microorganisms since von Leeuwenhoek time. Early 1700s. Why did it take, 100, 150 years for microbiology to really develop into a legitimate science? Well, there were a couple of reasons. One had to do with von Leeuwenhoek. Von Leeuwenhoek was very secretive, and he really didn't share his information probably as readily as he should. There was supposedly one instance where the Tsar of Russia came to visit von Leeuwenhoek and just was really excited to look through his microscope and see these little animalcules. He wouldn't let him do it. Okay, sorry, Tsar, you're not important enough. And after he died, interest in these microorganisms kind of died down again, and they really weren't thought to be of much significance. The real factor, though, that resulted in this lag period between the discovery of microorganisms and kind of the development of microbiology as a science had to do with the fact that during that lag time, they weren't able to do controlled experiments in microbiology. Now, you all know how important controlled experiments are in terms of the legitimacy of science, right? And what's one of the hallmarks of doing a controlled experiment? What should be the relationship between the experimental and the control? Should only be one variable, right? Only one difference. Because otherwise, how can you attribute an action due to the experimental if there is more than one variable in the two groups that you're comparing? Well, why couldn't they do controlled experiments? Because microorganisms in nature exist as mixed populations. And the techniques weren't available to separate bacteria from one another. Because to do an experiment that is controlled in microbiology, you've got to be working with what we call a pure culture. A pure culture consists of only a single type or species of bacteria. If you've got a mixed culture and you do an experiment with it, how can you know that the outcome is due to one species versus the other? So one of the things that had to develop in order for microbiology to become a legitimate science were techniques for separation of microorganisms, pure culture techniques. One of the other things that people realized about microorganisms is that they were everywhere. And that microorganisms served as contaminants. So even if you were able to obtain a pure culture, if you needed to transfer those organisms to a new growth media to keep them alive, how could you prevent contamination from occurring? So another technique that had to develop in order for microbiology to become a legitimate science were a series of techniques called aseptic techniques. These are basically sterile transfer techniques. So let's back up for a second. Just make sure you understand kind of where we're going. Legitimacy of a science depends upon the ability to do these controlled experiments and to have these pure cultures of microorganisms. But the techniques to make pure cultures just didn't exist during this period of time between von Leeuwenhoek and the mid-1800s. These are little tiny submicroscopic things. How do you separate them from one another? How do you transfer them from one growth environment to another without picking up the contaminants that are around you and filling your work environment? So without the development of these two particular techniques, pure culture techniques or separation techniques and aseptic sterile techniques, microbiology didn't have a chance. Okay? It couldn't be really a legitimate science. Well, it's not like microbiologists at the time said, okay, we're screwed. We can't be a legitimate science because we can't do this. We need to go out and develop these techniques. Instead, these techniques evolved as other types of investigations were taking place during that lag period of time between von Leeuwenhoek and the mid-1800s. In that particular lag period of time, there were two scientific questions that kind of dominated science. And one of those questions was, does spontaneous generation occur? Another question that simultaneously dominated science, and some of the same people worked on this question as spontaneous generation is, what causes contagious disease? So a lot of famous scientists were working on investigating these two particular questions. As people dug into and attempted to understand the answers to these two fundamental questions, techniques emerged from those investigations. Techniques that resulted in pure culture techniques and allowed separation of organisms into individual entities. Techniques evolved that involved sterile transfer techniques that prevented contamination. So these were the outgrowth of these investigations that were happening. And once these techniques were developed, that gave us the foundation for doing controlled experiments in microbiology. And then microbiology really did become a legitimate science. So what I want to talk a little bit about is, how did the investigation of these two fundamental type questions lead to the development of these essential techniques that were the foundation of microbiology? Okay? Does anybody know what the concept of spontaneous generation says? Ever heard of that idea before? Another word, a synonym for spontaneous generation is abiogenesis. What the concept of spontaneous generation says is that non-living matter can on its own become alive. Non-living things can spontaneously turn into living things. Is this a cornerstone of current biology? Amy, how you doing? Good, good. Welcome back. Stop by and see me sometime.
I will. What are you teaching right now?
Pardon me?
What is this class?
Micro.
Yes! I love micro. Just saying. Goodbye. Have fun.
Amy's one of our grads, and you're coaching now, right?
No, I'm athletic training.
You're athletic training.
Yeah, so my kids are.
Okay, cool. Yes, most definitely, stop by.
Come see it.
Okay. Today, the concept of abiogenesis, of non-living things all of a sudden magically becoming alive, is not something that we really take seriously. Instead, one of the cornerstones of biology, in particular cell biology, is biogenesis. Where do all cells come from? Other cells. Okay? Life begets life. Not non-life begets life. That's craziness. Okay? That's not something that you would ever think you would take seriously. Well, where did the concept of abiogenesis or spontaneous generation come from? Observations that weren't particularly careful. A pile of rags sitting in the corner, all of a sudden giving rise to mice. A piece of rotten meat giving rise to maggots. This idea that things that were alive or once alive could give rise to living things was fairly well accepted in times when people were a little bit less enlightened. And there was a certain comfort in that idea too. Because one of the things that abiogenesis depends upon is some kind of creative spark entering matter and causing it to become alive. Creation. A God. Okay? Well, one of the first controlled experiments that was done to deal with this idea of spontaneous generation and does it occurred on the macroorganism level. It was done by a scientist called Francisco Redi. And this guy did a very simple experiment where he was investigating this idea of whether meat could give rise to maggots, dead flesh giving rise to living organisms. And he took 2 containers with meat, and he covered one, and he left the other one uncovered. His hypothesis is that there was something, flies in this case, that were laying eggs on the meat, and that the eggs were hatching and growing into a larval stage, which is a maggot. And eventually those larvae would pupate into the adult flies again to complete the life cycle. And by doing something as simple as just covering the meat to prevent the flies from landing on it, you could prevent the maggots from hatching. So this was a very simple and elegant controlled experiment, which was pretty convincing evidence that spontaneous generation wasn't what was happening to cause maggots to form. And there were other experiments that were done, and kind of refuting spontaneous generation at the macroorganism level was fairly easy to do. But with the discovery of microorganisms, the controversy continued. When people first started studying microorganisms, one of the things they realized is that microorganisms grew pretty well in test tubes that contained water and then maybe some boiled meat or some plant material that was soaking in the water like straw. Typically these boiled meat solutions were called broths. Plant material that was left to soak in water was called an infusion. And one of the things that they noticed was that initially when you prepare these broths and infusions, held them up to the light, they were clear. But then if you let them sit for a while, they became cloudy or turbid. And if you looked at these clear and turbid solutions, there was one big difference. The clear solutions didn't have any of these little microorganisms, and the turbid solutions, they were teeming with microorganisms. So the spontaneous generationists said, aha, maybe spontaneous generation doesn't occur at this complex kind of macroorganism level. But we think it is happening at the microorganism level, this more kind of simplistic level of simple cells. And that non-living material in the broth or the infusion is giving rise to these living life forms. Well, people that didn't believe in spontaneous generation said, well, wait a minute now. We think there's something else going on here. We think that maybe these particular solutions are becoming contaminated. with either material that was already in the broth or the plant material. And the spontaneous generations would say, okay, here's what we're going to do to address your concern about possible contamination of the material used to make the broth. We're going to boil these things. So when we prepare this infusions or the broths, we're going to boil them. What should boiling do? That should kill any existing microorganisms that are present in the material used to make those solutions. And then we're going to set them aside. And when we do, they're going to get cloudy. What did the people who oppose spontaneous generation say? Well, okay, if you boil, you're going to kill the stuff that was already in there to begin with. But you're leaving them open to the air, and so if there are bacteria or other things in the air, airborne contamination could recontaminate these things again. And then what's happening is those contaminant cells are just dividing and growing and giving rise to the turbid solutions. Okay? So there were all kinds of interesting experiments that were done with boiling these broths and sealing the containers. Initially, they were using corks to seal the containers, but corks don't always fit really well, and they dry out, and sometimes they can allow contamination to get in. Some other experiments that were done were taking these tubes and drawing the neck of the tube out into a elongated tube to try to prevent contaminants from getting in from the air and traveling through the tube. And there were all kinds of weird and wild, wonderful experiments that were done that went back and forth trying to either refute or support the idea of spontaneous generation. At one point in time, the spontaneous generation folks said, well, if you boil these particular solutions, it destroys or injures the air that is needed to support spontaneous generation. There was really an interesting back and forth that one group would seem to refute a hypothesis and the other group would say, oh, well, but maybe it's this. Maybe you're injuring the air when you're doing your experiment of boiling these things. So a lot of different experiments were done with covered and uncovered containers, such as Rady did with the maggots, taking infusions and boiling them and leaving some uncovered and some covered. These researchers, Schultz and Swan, did some very interesting experiments where they took infusions and put little stoppers in them and little glass tubing in there. This particular one was very kind of creative. So here's previously sterilized infusions right here. This one had an open air inlet, and then the glass tubing was coiled, and then the tubing was connecting these particular infusions. They had a flame that was heating this particular coil right here. This was all done to address that concern that if you boil infusions, you destroy their ability to regenerate microorganisms because you're entering the air. Why did that experimental apparatus address that particular concern? Because when you boiled these infusions and then put these little stoppers and tubes in there, you had an open air inlet right here where fresh air could come in and basically replace the injured air that was created when boiling occurred. So we could get a new fresh air supply. But because of these twists in these tubes and heating this particular coil of tubing right here, any contaminants that entered this particular tubing would be killed before they came back in here and repopulated the flasks. Well, the person that really put this question of spontaneous generation to rest finally was one of the people who's considered to be kind of one of the fathers of microbiology, Louis Pasteur. Pasteur lived in the early to mid 1800s during that golden age of microbiology. Before Pasteur got involved in this controversy about spontaneous generation, he became very famous in France for another reason. He's credited with saving the French wine industry in the 1800s. One of the things that was happening in French wine in the 1800s is that there was a high rate of spoilage of the wine. Particularly wine becoming vinegary. And what Pasteur finally figured out was that the skins of the grapes not only had yeast, which would ferment sugar and produce alcohol, but that the skins of these grapes also had a bacteria contaminating it. And those bacteria would ferment sugar and produce acetic acid or vinegar and cause the wine to sour. So Pasteur developed a process that became the heart of the French wine industry for quite some time, whereby he mildly heated the grapes to the point where it killed the bacteria but did not kill the yeast. And consequently, sugars that were fermented then were turned into alcohol and not into acetic acid. That particular process of gentle heating to kill some organisms and not the others is called pasteurization. We'll talk about pasteurization later on. So he developed pasteurization. Pasteur went on to, after he was involved in kind of refuting spontaneous generation, was involved in investigations of the germ theory of disease. He developed a rabies vaccine. He discovered the bacterial cause of cholera. So he was a real giant in microbiology. Here's Pasteur's experiment. Pasteur used broths, and instead of sealing those flasks with either corks or rubber stoppers or something like that, he had a glass blower heat those particular flasks and then draw out the neck of the flask into this elongated tube, which was open to the environment. These are called swan neck flasks. He vigorously heated the broth to kill any contaminants that might be there. And then he just let those flasks sit. And no matter how long he let them sit, they didn't ever become cloudy. The fact that these were open addressed the concern that spontaneous generations had about him injuring the air as a result of vigorous heating and boiling. There are Pasteur flasks still in the Pasteur Institute today that were made in the 1800s by Louis Pasteur that still have clear broth in them that have been sitting there for all that time. If he broke the neck of that particular flask and contaminants could get in, it quickly became contaminated and cloudy. If he tipped the flask so the liquid could go into this particular extension of the swan neck where contaminants might have entered, and then tipped it back again so he washed those back into the flask, kind of like backwash when you're drinking, okay, they again became cloudy. So this particular kind of simple, elegant experiment done by someone who had a lot of legitimacy, Pasteur, was really kind of the death knell for the idea of spontaneous generation. There were a few little hiccups here and there. One of the hiccups was sometimes people who used broth cultures in their Pasteur flasks, everything worked out just right. Some people who were using infusions of plant material like hay, the experiment didn't work. And one of the things that later was found out was that plant material was contaminated with bacteria that can produce structures called spores. And these endospores were heat resistant. And that boiling wouldn't kill them. Really, the only way you could kill these endospores was to boil under pressure. And so the technique of autoclaving arose as a result of this particular realization. So as people worked on this idea of spontaneous generation, a couple of things emerged. And that is, there are bacteria everywhere. And they're going to contaminate things if you let them. So as people worked on spontaneous generation, aseptic or sterile techniques developed to try to prevent contamination in these particular experiments. And techniques like autoclaving and sterile transfer and things emerged from this study of spontaneous generation. But these particular studies had nothing to do with the second cornerstone, getting a pure culture, separating microorganisms from one another. Those techniques developed from a study of the second question at the time, what causes contagious disease? So that's where we will go on Friday, is we'll talk about the study of what causes disease and how that led to the development of pure culture techniques, which ultimately gave us the second bedrock for the development of microbiology as a science. So let's take about a 10-minute break. Reconvene in here at 11. Make sure you sit where you want to sit for lab, and we'll go through some lab orientation.